Now the second part of this video, we're going to look at the active transport processes. And active transport is the movement of substances from low concentration to high concentration. And there's two basic types. So why don't we pull this up? Okay, so here we have basically what we did last time. Passive transport is going to be on your left-hand side. Let me just make sure it's working. Is on your left-hand side over here. Okay, and that's going to be the simple diffusion, facilitated diffusion, filtration, and osmosis. So active transport on the other one that we're going to be looking at is going to be using energy. So uses energy. And that energy comes in the form of ATP, adenosine triphosphate, okay? And there's two primary methods, okay? There's going to be primary transport, or primary active transport, if you will. And primary uses ATP to move substances from low to high. So low to high. Okay, so that's all that basically is. And the easiest thing of think of pumps. Okay, so you have a sump pump in your basement. So if water gets into your basement, there's more water on the outside than in your basement. So you take what is collecting in the basement and you pump it back outside. I'll have a couple images here in just a second to go through this again. The second one is going to be secondary active transport. And secondary active transport, what we're looking at on this one, it's using energy from movement of substances from high concentration to low concentration. So think of kind of simple diffusion. Think of sodium moving down concentration gradient. Now, though that is passive energy, it's not using any energy to take sodium from a high concentration to a low concentration. But that movement, that offers a potential energy. That movement is potential energy, and it's converted into kinetic energy. So as sodium, typically sodium, as sodium moves through the channel, it provides energy to then power that protein to move a substance from low concentration back into high concentration. Okay, so think of water in a dam. Okay, so water flows over a dam. Well, that's passive movement. As the water fills up in the dam, it breaches the surface, goes over the dam. That's very passive. Okay, but how do you generate electricity from that? Well, that passive movement of water going over the dam then powers or turns the generator to then form electricity. That's kind of what we're doing here. Instead of water, we have sodium that is generating energy in order for a pump to work. Okay, so you're changing potential energy into kinetic energy on this one. And there's two types of secondary active transport. Type 1 is a Simport. And we're going to show images of these. And that's moving two substances in the same direction. So you got a pump, and let's say sodium comes in on one side, another substance follows that through to go to that side. Okay, so that's a Simport. They are basically piggybacking its way in. And then the other one is an Antiport. Now, Simport is also a co-transport. I think it has another co-transport is one. Coupled transport. Contra-transport is going in opposite directions. Counter transporters or antiporters. Don't worry about those words right now. Antiporters are something goes in this direction, providing the energy to move something back in the other direction. So let's look at a couple of quick images here. Let me hide this, and let's look at a couple real quick images. So this first one here is a basic pump, okay? So these are calcium ions. Okay, how do I know they're calcium ions? Well, it says right here, calcium, those are ions, okay? The +2, think about what that +2 means. See if you can figure that one out. Basically, we have high concentration on this side, we have low concentration on this side, but the calcium is going into this pump and it's being pumped out, okay? But it's using ATP, okay? So this is a basic pump. The other image we have here, this is a little bit better for learning the sodium-potassium pump, okay? And sodium-potassium, we will learn that sodium is your primary cation on the extracellular side of a cell. Potassium is the primary cation intracellular. When sodium enters the cell, and we're messing up these balances, okay, sodium enters in, potassium rushes out. Now we have to reverse that process. We want to get the sodium out and bring the potassium back in. Now, throughout the semester, we're going to really talk about this. But here, what this is basically referring to on here, we've got a pump. The pink structure here, that's a pump. You've got 3 sodium from the intracellular space, binds with this protein. ATP is used. It changes its shape, and the sodium leaves. Potassium then comes in, changes its shape again, and potassium is released inside the cell. That's a basic pump, but there's it uses ATP in this case. Okay. The other thing I want to look at, let me just see what we want to do here. Oh, the Simport and Antiport. I think I have that on another image. Let me look at this. Okay, here's a Simport and an Antiport. Okay. Now remember, this is a secondary active transport method, meaning you have the passive movement of one substance, say sodium, okay, moves through a channel that provides the energy. Remember, water going over the dam provides the energy, makes the electricity, okay, and it allows another substance to be actively moved. Actively meaning requires ATP. Now there's symporters, and symporters, I said 2 substances move in the same direction. Now these purple things that you see here, those are representing sodium ions, okay? Now we know sodium moves through a channel passively down its concentration gradient. So if you look at the number of sodium up here, we have far fewer down here, so sodium is passively going this direction. That movement, think of these as the water, okay, going over the dam, that provides the energy source for glucose to then enter the cell, okay, because that requires energy. So they're both moving in the same direction, therefore it's now called a symporter. An antiporter is going to be this other one, okay. Now again, sodium here, higher concentration external or in the extracellular area than you do on the inside. There's a passive movement in for this one. That provides the energy, but here now we have hydrogen ions. We have low concentration intracellular, higher extracellular. That requires energy to leave the cell, so the sodium coming in is providing the energy for these to then come out. And that is now an antiport in this case. Okay. Now let's look at one more here. I think we've got let's do this one. Okay. Now let's look at a couple other types of movement across the cell membrane, okay? And this can be, the first one is a vesicle. Remember last semester when you were talking about the various organelles inside of a cell? A vesicle is just a membrane-bound sac holding something. So think of a water balloon as a vesicle. The balloon itself is the membrane, and it's holding water. A vesicle is a membrane holding something, okay? And as you can see on this one, let me get my glasses on so we can see. So we're intracellular here, intracellular, and this is extracellular out here. What we're doing is, this is membrane, okay? Now this membrane, this is your bilayer, okay, is made up of the same thing, different colors, but it's made up of the same stuff. As you can see, it merges in, and when it merges in, when it fuses to this membrane, it opens it up, and then this stuff comes out. Well, that whole process of moving around requires energy, okay? So this is basically what we're going to call exocytosis. E-O-S-I-S, exocytosis. Substance from the inside is being moved out of the cell. Opposite of that will be endocytosis. And endocytosis is going to be this one, okay? Endocytosis is movement of something from the outside into the cell. So this is endocytosis. And there's a few different types of here. So here we have phagocytosis, okay? In basic biology, you may have learned about phagocytosis. And what it is you have a substance out here that the cell wants to eat, and the amoeba or whatever it is kind of works up to it, and then it starts to engulf it. Okay, that requires energy. It starts to wrap around it. And once it's engulfed, that's what we have on this one. So basically, this is this top picture here is kind of a stop motion one. So here's the stuff that the cell wants to eat. You can see that the cell is starting to go around it and to engulf it. Now, if we were to go to the next stage, I'm going to do this in red. This one kind of goes this way. This one goes this way. Okay, so that would be a representation of the membrane, and it pulls it in from here. Okay, so the movement of the plasma membrane around this requires energy, requires ATP, and now you bring that in. That's phagocytosis. Okay, pinocytosis is going to be this other one over here, P-I-N-O, not P-H-A-G-O, but P-I-N-O. The way I remember this, I can't get it out of my head if I try to. Pino sounds like wino. Wino is a liquid. Okay, wine, you're drinking liquid. So pinocytosis, it's called cellular drinking. Phagocytosis, cellular eating. So phagocytosis, you're taking a solid substance in. Pinocytosis, the cell's taking a drink of wine. All right, pino, wino, okay. I know it's weird. So it's the same thing. The cell membrane kind of wraps around some liquid and pulls it into the cell. That's all that basically is. The receptor-mediated one is a little bit more complex down here, and that deals with these little green things. Those are called catherines. Don't worry about that so much, but we have receptors on the outside of the cell, and when something binds with it, whatever that receptor is sensitive for, whatever binds with it, then those receptors and these calthrins they're moving across the membrane of the cell. They accumulate, and when they accumulate, they engulf the stuff on the inside. So this is going to actually go that way. This is going to go this way. So these calthrins, let me do a green one on this one. you're going to have a little thing here, and you're going to have little things, but those move in this direction to form this network around this vesicle, if you will. So it's a calthrin-coated vesicle on this one. And then once this moves into the cell, then those calthrins release, and then the cell can utilize that stuff for whatever it needs. That's a little bit more complex than what we want to get into for this purpose, but for active transport, we have the requirement of energy, ATP. Okay, there's a primary and a secondary method. Primary just uses ATP to power it, like a pump. Okay, the secondary uses the kinetic energy associated with, let's say, simple diffusion or facilitated diffusion. Okay, well, facilitated diffusion. So it's typically sodium. So as sodium enters through the channel, that powers the channel to then pump something either back in the opposite direction, which would be an antiport, or the movement of sodium coming in powers the pump to then pull something else in. And what we saw earlier, glucose would be associated with a symporter, hydrogen ions with an antiporter. Okay, and then we have the two different types of movement of bulk stuff around, and that's going to be exocytosis, which is basically waste products in the cell that the cell wants to get rid of. So the cell's just pooping it out. Okay, so that's a form of exocytosis. And endocytosis, there's your three versions of that. Phagocytosis, which is bringing in, say, food, a large solid particle of something. Pinocytosis is cellular drinking, so it's bringing in liquids with it. And then the receptor mediated, basically you've got receptors on the extracellular surface of the membrane. Something binds with that. Those receptors kind of then move around the cell membrane. They're not fixed, they're moving around. And they accumulate, and the calthrins on the intracellular side form this vesicle, and it pinches off, and then it pulls it in. That's as far as I want to go on that. So that is active transport, and I think, if I'm not mistaken, we have cellular respiration. That's a big one. So stay tuned.